Okay, thank you to each and every one of you being here with us again. UA World Innovation Summit was founded by students from 8 top universities around the world, including Oxford, Cambridge, Harvard, MIT, Stanford, Caltech, Tsinghua, and Peking University. So I'm Yongchen Jing, a PhD candidate from the Oxford University and the co-founder of UA Summit. Usually, a compere will be here to host event and give introduction. Today, I'm very honored to jump in and make an introduction to the Professor David Sinclair, as I'm a big fan of him. Professor David Sinclair is an Australian-born biologist. Well, coincidentally, I'm also did my bachelor in Melbourne Uni, Australia. He's a professor of genetics and co-director of Paul Glenn Center for the Biology of Aging at Harvard Medical School. He's known for his research on aging, and he has received numerous awards for his research. In 2014, Sinclair was included in Time 100 as one of the 100 most influential people in the world. And in 2018, he was included in the Time magazine 50 most influential people in healthcare. Professor Sinclair's research involved a very important protein called SIR2 and its cofactor NAD+, which are proven their activity reduction played a critical role during our aging process. His most recent paper, published in Nature, gave an amazing example that rodents are able to recover useful epigenetic information and restore vision by ectopic expression of 3 Yamanaka factor, OCT4, SOX2, and KLF4, also called OSK factors. As the author of the bestseller book, Lifespan, Why We Age and Why We Don't Have to, released in 2019, if there is someone who able to treat the aging as a disease and extend our lifespan in this century, David Sinclair must be one of them. So please let's welcome Professor David Sinclair to share his view on aging.
Thank you, Yongchen. I appreciate it. Welcome, everybody. Good morning. Well, around the world. Let me, so I'll just share my slides now. Yongchen, is that?
Yeah, sure.
There you go. All right, let's start with that. Okay, hopefully you can see this. Are we good to go?
Oh, good.
Good, all right. So I'm gonna talk for about 40 minutes and then we'll have questions. So why study aging? Well, I've been fascinated with aging since I was 4 years old. My grandmother told me about this thing called aging and that everybody gets sick and dies. And it surprised me that no one was talking about it at the time or ignoring it. And we tend to do that because it's a tough thing to talk about and think about. But we do need to address it. If not for social reasons and health reasons, then for economic reasons. Because since 2018, for the first time in human history, the number of older people now outnumbers younger people. And so the goal here really is not to make people live forever. That's absolutely not the right thing that drives us. What drives us actually is the thought that we could tackle numerous diseases with a single treatment. It could be a pill, it could be an injection, it could be other things that monitor our health. We have the biosensor revolution right underway now. And I do see a future where we will not leave the house, or we wouldn't dare walk around without being monitored for something that could happen. My research really is based on the idea that we have inbuilt protective mechanisms against aging, and we can turn those on. The sirtuins, which Yongchen mentioned, are a set of genes among many that control how long we live and how healthy we are in old age. And we've known for many years that the majority of the health in our old age is not genetic, it's epigenetic, which is the basis of a lot of my research. The sirtuins are genes that control the epigenome. So I'll explain what that is. But before I leave this slide, I want to leave you with one more thought. And that is that for the last 200 years, we've made great gains. We've pretty much done all the easy stuff. We've tackled heart disease very well. We've done pretty well with cancer, and we're not there yet. But the problem with addressing one disease at a time is that as you get older, the chance of these diseases goes up exponentially to the point where even if you cure cancer, you're only going to live on average 2 years longer. And that's a problem, right? Because that means that we're not going to continue to make these gains in lifespan. We've done all the easy stuff. We've kept ourselves healthy pretty much until the age of 80, if you look after yourself. But to go further than that, we need something different. And so I'm all about keeping the body, every organ, not just one organ or one system, healthy. And that way we'll have these enormous gains. But also we won't have older people who are sick and say have dementia for the last 10 years of their life, because while we can keep the heart healthy, we have not been successful keeping the brain healthy in old age. So hopefully I've convinced you this research is worth it. And one of the things that struck me years ago, and I've been talking about it, writing about it, is the crazy notion that age-related diseases, which cause 80% of the world's suffering right now, are in a separate box to a separate circle to aging, when actually, in reality, what's going on is this. Age-related diseases are the manifestation of aging, right? You don't get heart disease or Alzheimer's when you're 12. It sounds obvious, but the reason is that the body is resilient and can repair itself when you're young, but it cannot when it's old. And why is that? That's one of the questions that I've been trying to answer. And if we can slow down or reverse aging, then we won't get those diseases. We won't have to even treat heart disease if we are able to keep the body young. So I'm going to talk today about our research in the lab and others around the world, trying to understand what is aging, what happens at the molecular level. I'll tell you how we now can quantify it. I can show you that we can slow it down. And as we just recently published, it does appear that you can safely reverse aging in a complex tissue such as the eye in mice. And we can do this age reversal in human cells, human neurons, and other cells, fibroblasts, skin cells. And so I think that tells us, at least in mice, but there's no reason why it wouldn't be the case in humans, that aging can be reset. The age of the body can be reset and safely. That's the important thing. And it's also can be reset in vivo. All right, so there are various stages of aging, and I've classified them in this review that we wrote as development, which is A0 up to A1 when you're born and you're healthy. And this would be up until age 20 to 30. Very little goes wrong with you typically, because your cells have resilience, your body has homeostasis. And even if you get injured, the body has great capability of healing itself at the molecular level and at the tissue level. But after that, you tend to get less and less resilience and less healing. And eventually your body can't even keep diseases at bay. And this is the aged phenotype, we would call it, the aged symptoms that we all know too well. And this is what eventually kills us, most of us. One thing that's interesting is that cells, if you regard these as stages of the cells in your body, there's this, what we think is a terminal stage called senescence, which is when a cell checks out of cell division and sits in the tissue, causing havoc. Senescent cells are in our bodies. We can find them. They're not too hard to find, but they leach into the bloodstream factors that cause inflammation and cancer. And so there's a push to kill these cells off with drugs and molecules called senolytics, which kills them, or xenomorphics, which alters their behavior, so they're not toxic. I'm not talking about this today so much. I also have spent a lot of my career working on these sirtuins. Here's SIRT1, where there are chemical activators. Resveratrol was the one that got all the attention about 12 years ago. We work on others now. We have much better molecules that are 1,000 times more active. They extend lifespan in mice, and we're in clinical trials now with these other molecules called NAD boosters, which also activate these sirtuins that protect the body in various ways. You can fast. I'm pretty well known for someone who likes to skip a meal or two a day, because it's very clear that this is a way to activate the body's defenses against aging and we think slow down that process of aging. There are other drugs that are even on the market that are suspected to have an effect on aging. Metformin is a diabetes drug, which a few people take. And rapamycin is not one that you would take, in my view, certainly not in regular doses because it has toxicity. But it's used in the treatment of transplant rejection, cancer. It's useful, but for aging, it's really not yet proven out. But what's good about it is both these molecules will extend lifespan in mice. And looking at the effect in older people, both of those drugs seem to have benefits, various ones that I won't go into. But what I'm going to talk about mostly today is this ability to reset the age of a cell or a tissue. Now, we don't want to go too far. If we overdo it, we could cause cancer. Cells that are embryonic will just grow out of control. That's not the goal. We want to take their age back partially and then reset them so that the tissue can go and age out again. I'll just give a few slides on that. Hormesis is the term that basically describes whatever doesn't kill you makes you live longer. You want to put the body in a state where it's sensing adversity. You don't want to overdo it. You don't want to harm the body irreparably, but you do want to put the body in a state where it feels uncomfortable. So how do you do that? Well, so this is high intensity interval training up here. So lose your breath, get on a treadmill. That's pretty easy. Skipping meals, put your body in a state of fasting, triggers these things. We think temperature might do that. There's some good evidence, certainly in saunas, that they're good for you for reasons that we don't fully understand, but it might be hormesis. Low amino acids, so eating plant-based food that don't have huge amounts of leucine and isoleucine. These are branched chain amino acids. Valine is one. And then we've got xenohermetins. You've probably never heard of those. These are molecules that plants produce when they're stressed and under adversity, such as resveratrol is the most famous one, but there's many others, quercetin, fisetin. Plants make these to survive. And we have a hypothesis that we published in Cell a number of years ago, that when your food supply is under adversity, your body wants to know that. These activate the sirtuins, and also can kill off senescent cells in the case of fisetin. So there are three main pathways that in the body that regulate aging. There are many other proteins that I don't have time to tell you about, but these are the main ones. mTOR in general senses fasting and low amino acids. AMPK senses low energy. You're not eating enough glucose. And the sirtuins will respond to all of these things. And together, they do various things. I mentioned they prevent senescence or kill the cells. They remove the unfolded proteins from your body, so they should be helpful against Alzheimer's, for example. They will protect the telomeres, which are the end of the chromosomes that get shorter. They will preserve the health of stem cells, which you need to stay young, and they'll also improve the body's metabolism, so you don't get or have such a predisposition to type 2 diabetes. And there's a long list of what they do. But in general, when we do this to an animal, a mouse or a dog, and treat them with these molecules, what we see is that they have improved health. And if you're healthy and you're fit, you don't die, generally, right? So longevity is the outcome of that. And we can extend the lifespan in the field somewhere between 10 and 30% pretty easily these days. But the question is, can we do this with people? Can we take a pill that would mimic all of these things? Because you can't expect an elderly person to go run on a treadmill or even to go hungry. So that's why I want to make medicines that do the same or better. So I mentioned resveratrol. That's not a drug. That's just a supplement. We have made molecules that went into humans that worked well in psoriasis. But one of the things that I'm particularly excited about is this NAD plus molecule that Yongchen mentioned. Sirtuins need NAD to carry out their enzymatic reaction. And as we get older, we make less of it. And so if you look at the skin of a person my age, I'm 51, we would on average have half the levels that we had when we were 20 of NAD. And therefore our sirtuins are not working actively as they once did. Now, when you run on a treadmill and you're hungry, there's good evidence that your NAD levels will go back up, okay? But perhaps we could supplement an animal or a human with an NAD booster, a precursor to NAD, such as NMN, or there's one called NR as well. You're probably wondering what MUFA is. That's monounsaturated fatty acids. Oleic acid was just shown to activate one of the sirtuin enzymes the same way resveratrol does, by binding into a pocket that's on the N-terminus for the biochemist. And then, so let's say that's the N-terminus, these molecules bind, and that comes in, and now you activate the enzyme. And we can activate the enzyme, at least in vitro, more than 10 times. All right, so how do we know that these molecules are activating sirtuins? Well, we've made a mutant mouse that is unable to be activated because we block this elbow. And so now you don't get this action. And now we feed that mouse resveratrol, and we're finding that it doesn't live longer in the case of high-fat diet, among other things, doesn't run further and all that good stuff. But I'm not talking about that today. I just wanted to mention it. I want to get quickly onto something else. But before I do, we put out a paper, this was 2018, and we found that if you give old mice, so 20-month-old mice, NMN in their drinking water for 4 weeks, they can run just like a young mouse. And the main reason for that is that they have more capillaries or capillaries. They just have better blood flow. And we also showed this in collaboration that it works in the brain as well to improve blood flow and regrow the blood vessels in the brain in a healthy way. And that was Zoltan Ungbari's lab. So hopefully you can see this black mouse on a black treadmill, the one that was drinking NMN in the water clearly has better stamina. It was night and day. They could run 50 to 100% further than the untreated mice. And so as I mentioned, we're doing clinical trials. We have labs all over the country, hospitals, over 30 hospitals that are part of this program to test this in COVID patients and also in elderly patients that have frailty. And we're also treating a rare disease called Friedreich's ataxia down in Pennsylvania at Children's Hospital there. So we'll know in the next year that whether or not this has similar effects to the mice. We're looking in collaboration with Shelley Basin. He's at Brigham and Women's Hospital at Harvard. He's the one running that study. And so they're going to look non-invasively at the bioenergetics, production of NAD, and even ability to cycle inside an MRI that's measuring these things. So that's where that's going. But let's now talk about one of the big breakthroughs in the lab, which is epigenetic reprogramming. So the first question really is, what is aging? And what I think it is, it's, if you boil it down to its essential basis, you can write a mathematical formula to describe aging. And that's the loss of information. Okay, and Claude Shannon, who was at MIT during most of his career, was fascinated with information preservation, because in World War II, people would lose their lives if a radio signal didn't make it to where it needed to go. And he came up, the black lines are stolen straight out of his paper from 1948. And it's a landmark paper that actually gave rise to the entire internet. And what he said was, there's a transmitter of information, and this could be a radio signal, could be, an email now. And he said, we always have noise, especially it's even worse when you have an analog system, but even digital will lose some information. And then if you lose that information, you can have this system that goes back and gets the missing bits from the observer. Now we'd call that the backup copy or TCP/IP protocol. And then you use that to correct the data so that this information is identical to what it was earlier. Perhaps, go back to here. So some of the questions that we have in my lab is this aging? And I think it is. what's the source of the noise, and can you reboot the system and get the information back to be young again? And so what we used in our lab was cells, of course, but we also used the eye as a model system to reverse the aging process in a mouse. So one way to think of this also is that there are two types of information in the cell in our bodies. There's the digital information, which is the ATCG, 4 letters in DNA. That's our genome. And we do lose a bit of that information, but actually what we found in my lab is the majority is the other type of information that gets lost during aging. And that's the epigenome. Okay. In the case of this analogy, it's the epigenome is like the reader of the DNA. It controls which songs we play in our cells over time. And aging, I think, is like scratches on this CD. I think most of you know what a CD is, even if you haven't held one. You know, maybe you could think of aging as the spinning disc in a phone, if you've got an iPhone, or a blue screen on a computer that needs to be reset so that the software can be reset and then read the information correctly. So in biology, what we're talking about is the DNA is digital and the epigenome is generally analog. For the most part, the epigenome are the control systems, these loops and these bundles of proteins that hide the DNA from the cell. We call this heterochromatin. And this was a proposal that myself and Shin Imai and Kitano, back in the late 1990s, came up with this. Lenny Guarenti was our supervisor at the time. And it stemmed from the discovery that sirtuins can regulate yeast aging and make them live longer. And sirtuins, the SIR, stands for Silent Information Regulator. So we knew back then in the mid 1990s to late 90s that information was key. But actually, to be honest, the sirtuin field moved on and forgot that information was important. In my lab, we've been working on this ever since. And so really what I'm saying is that when cells lose this epigenetic information, genes that were once on get switched off and vice versa, leading to a loss of cellular identity in an old cell. And this is what I think is aging. And of course, there's a lot of other things that lead to this, or that are a consequence of this, such as inability to respond to damage, inability to get rid of misfolded proteins. And it's my belief that if we can reset a cell, hopefully all effects of aging will go away. So what drives aging? Let me tell you about that. I'll quickly go through this because I'm going a little slower than usual. This is how DNA is packed. This is what it really would look more like if you were inside the cell. These are the histone proteins and the DNA is in blue. So digital is blue, analog is this folding pattern. And there are these little tags that are put onto the DNA as well as the proteins. And one of them that's really important to control gene expression and cellular identity during development and birth is DNA methylation. Okay, and so imagine a little chemical here, a carbon with 3 hydrogens on it, that gets stuck on DNA and can actually last your whole lifetime to say, all right, this DNA encodes a gene that is liver specific, and now I'm a brain cell, don't ever turn it on. But during aging, as I mentioned, there are changes to the epigenome, including changes to DNA methylation, that we can actually read and use it as a clock for aging. Stephen Horvath gets the award for being the discoverer and the promoter of this DNA methylation clock. And what we do now, based on Steve's discovery, is we can read the entire DNA methylome, as it's called, and we can plot the key parts of that we train using machine learning. This is the training set to build this algorithm. Then we can use it as the test set to predict the age of a tissue or a person or a mouse. Steve's done hundreds of species now, and it's a universal property of life that these DNA methylation clocks can predict age and also future health. So actually, you'll notice that there's also aging going on during development. So if you think that if you're 20 years old, you're not aging, think again. It's a constant process. And one of the drivers we found in yeast originally and now in mammals is DNA breaks. And we think that DNA breaks are particularly toxic for the cells because they distract proteins such as the sirtuins from where they should be and make them go to other places. So eventually the epigenome is messed up, and that this is a process that drives aging. So if this is true, if we slow this process down, we should extend lifespan. And if we speed it up, we should accelerate it. So we did the latter. We did this in yeast years ago, but doing it in a mouse is at least an order of magnitude harder and longer. And so we made these mice called ice mice back in 2008, and been working on them ever since. And we can take the mice and make them have an aged epigenome. And now we can ask, do they get older? And we also can use the cells from these mice, which we've been doing as well. And the question is, does the response to DNA breakage induce changes to the epigenome or chromatin that drive aging? So just very quickly, I'll go fast because I want to get to the reprogramming stuff. This is work by Jae-Hoon Yang in the lab, who shows that if you create cuts in the genome of a cell and have them repaired without any mutations, no changes in cell cycle, that there is a loss of epigenetic information, that this cell here, even though it has the same genome, it's not the same cell that it was here. There's a lasting scar on the epigenome. So what are those changes? And if you're wondering how many breaks do we induce, it's only a few cuts per cell. It's about 3 to 4-fold above the baseline. So this would be, if this was 1 cut per cell, this would be 3. So it's not a lot. Typically, people who study DNA damage create thousands, if not tens of thousands of cuts or sites of damage. So this is tickling the chromosomes, but it does lead to some major changes. We see changes in development. All of these are, maybe not all of them, but these are the top 20 processes in the gene sets that are altered. If you look at the chromatin changes, so these are methylated histones, we're seeing Hox genes. So what are Hox genes that change? Well, these are genes that you don't want to mess with typically. These control your head from your tail during development. And what we see over time, this is just a way of representing these changes across these Hox genes, which are in a cluster. We see that on this left-hand side, these genes go down in the levels of expression. And on this side, they actually have higher expression. And this is very 1- and 2-dimensional, but we can actually now go in and look at 3 and 4 dimensions of the genome. The way we do that is a process called ChIP, chromatin IP, and we use this, we're using a special type of chip, which is called high chip, where we can look at specific loops of DNA that have different modifications on these histones. And this is a acetylation of the histone, and down here is the methylation. And one of the things that I hope you can see is that, so this is representing a loop where DNA is touching another gene, right? So up here, we've got a little part of this cluster touching this promoter. This is a poised promoter and a poised enhancer, which is not functional. But after the aging process that we induce, this ICE process, now you can see there's a lot more effects on these bridges here. Okay, so this is changes in the 3-dimensional structure of the cell that lead to changes in gene expression. And we've done this across the whole genome, and the whole genome has these hotspots that, like these Hox loci that change. Now, we don't know why these particular ones change. We're not cutting anywhere near a Hox gene, but there are these distant changes. But now what's exciting is we can do this in a mouse, the poor little mice, we do this too. What we do is we treat them either with just control mice, we treat them with doxycycline, and these are the controls that look like this 10 months later. So this is the unscratched CD. And then here what we see is a mouse that was treated 10 months earlier that's now become what looks a lot older, a scratched CD as it were. Now we've looked at these mice for 10 years now. We haven't even published them yet, but we're trying. We can look at their organs, and you have to trust me, actually, all of their organs look old. We can also use a more quantitative way to look at their age. We can do this DNA methylation Horvath clock on them. And as I mentioned, most animals and people will be along this straight line, But if you've aged them prematurely, they'll be up here. And if they're healthier, if let's say that mouse has been only eating once per day, it will literally be younger based on this clock. So we expect that these mice should be older, right? If we're true, if our hypothesis is correct, they should be older. And we were excited to see that they were older based on, so the red dots are these ice mice. And they're about 50% older on average. Not all of them, but most of them are. So what we want to do now is see if we can reverse aging. Now, just one thing to remember, there are these TET enzymes that remove the methyl off the DNA. So if we're going to be reversing the clock, it's probably going to require these TET enzymes to remove those. And we test that later. All right, so is there a reset switch? In other words, can we take this messed up epigenome and reset it? It's a lot to ask. It means that there must be, if this works, there must be a memory, a backup copy of a youthful epigenome. Something that no one has ever discovered, but there are hints that it exists because it's possible to reset the entire age of a cell. And you can do that either by nuclear cloning, and now you get a new tadpole, or you do cellular reprogramming, which is using 4 Yamanaka factors here, OS, K, and M. And you can, and these two discoveries led to the 2016 Nobel Prize for John Gordon and Shinya Yamanaka. There are other reasons to believe that you can partially reverse aging without going through a single cell. It's great that you can reverse aging or go to zero, but it's not much use if you're already an adult. So you want to be able to reverse it partially, and it looks like we might be able to do that. So Hydra can live forever, or essentially forever. Axolotls grow new limbs. We even grow new livers. One of the parts of the body that very early in life loses its ability to regenerate is the central nervous system. And we know that if we break our spine, or if you lose your vision and damage the optic nerve, it will not grow back. But in an embryo, it will grow back. And in fish, adult fish, it does grow back. And so if we want to take the adult nerves that are from an old mouse or an old human, we used human nerves, and we want them to grow again. And so the student Wencheng Lu was working for years trying to find a set of genes that would partially reset the age of a cell without making it cancerous. Now, if you use the Yamanaka factors, they will become a tumor. This is the problem. You don't want c-Myc in there because that's an oncogene. But the question was, if you do this, what happens in an animal? So Alex Ocampo and in the Belmonte lab at Salk Institute, they made a mouse and published in 2016 that if you turn on all 4 Yamanaka factors, yes, if they stay alive, they'll get tumors. If you turn them all on at high levels, they will actually kill the mouse after 2 days. So this is all not looking very good, but Alex took it one step further, and after 2 days, turned the genes off, and then waited 5 days, turned them back on for 2 days, and cycled that. And in a prematurely aged mouse, there's one called the lamin A mutant that in humans leads to the children who look old. What he found was that those mice did a lot better. In fact, they lived about 30% longer. Now that was remarkable, but it's certainly not going to be a therapy because it either kills or causes cancer. So Wencheng Lu decided he was going to have one last ditch effort to reverse aging partially. And he put 3 of the Yamanaka factors into human neurons and skin cells and found to his delight that it worked. And so the system that he built was this TET on and TET off system. So he could turn these genes on and off at will in cells. And he found that even though age went backwards, he could maintain the cell's identity. They didn't induce the downstream tumor or oncogenic pathway. It was safe when delivered either in the whole body of a mouse or in the eye for over a year. And he used an AAV, which is already used in gene therapy, as I'm sure you know. In the eye, it corrects genetic diseases. There's CRISPR on the way to replace those genes and fix them. So what we did in collaboration with Zhigang He's lab was to try it in this nerve crush model at the back of the eye. And then we can put a dye in to see what color and how far the optic nerves go back to the brain. And then we can also have mice where we turn on the OSK genes with DOX, and then we get to see whether the optic nerve is younger and if it grows back. So the eye is here, the brain is here, and you can see that these nerves here have died. That's typically a response to damage of nerves. And a lot of the nerves here have died off too. You can see there's empty spaces. But if we turn on the reprogramming system just after the damage, this was the first result we got, that there was a lot of preservation and they started to grow back to the brain. So this is just after a couple of weeks, but we've also got, we left it for 4 weeks and 16 weeks and found that those nerves, many of them grow all the way back to the brain, which is off the screen here. And you can quantify this. And essentially what this says is that the 3 gene combination is the only one that works. Singly, they don't work. Put in different viruses, they don't work. You have to put them all in the same virus. And then we asked, does it work, or does it affect the age of the cells in the eye? And the answer was yes. They are younger. Interestingly, we could see that age was advanced by the damage. Who knew? We didn't, that damage was also an acceleration of aging. But here's what's really interesting. This is just the clock looking at a few hundred sites. But now we can look at all of them across the epigenome. These are DNA methylation sites. And you can see this is an intact, undamaged. This is the injured optic nerve after recovery. It's not the same. A lot of the cells have changed their methylation pattern in a way that resembles aging. And the OSK maintained that, or reversed it, you could say. And this is another way of plotting that. This is injury versus aging, and the correlation between the two is huge. Now, what about these tests? Are they required? And the answer is yes, you do need the enzymes that remove DNA methylation for this to work. And as I'll show you later, you can also restore the vision of mice that have had either glaucoma, pressure in the eye, or just old mice. And this was the old mice. You can see they get better at vision with our treatment. And it literally requires the DNA methylation process in the eye to restore the vision. So about the glaucoma, anyone who's had a relative with glaucoma knows that there's nothing you can do to reverse this disease. All you've got is pressure relief, which is not good enough. We want to be able to restore vision in people who have gone blind. So that's what we tried. In this case, it was a mouse model. We put beads in. This is in Bruce Cassander's lab. You can, he's at Mass Eye and Ear in Boston. And now we can turn this on after the pressure has ruined the retina. And look at what happens. 4 weeks later, we get a restoration of electrical signals in the back of the eye. So now it's back to normal. And if we look at mice that are looking at a screen, if they move their head, that tells us they can see this moving object, these moving lines. And that's called the optomotor response. And if you graph that, you can see that the treatment partially restores the vision in these mice. This was 4 weeks. Now we've done 8 weeks, and we get full restoration of vision on average in these groups of mice. So this is the first time that's ever been possible. But what about old mice? So ignore the feces here. They're just scared. But this is an old mouse that is not turning its head. It's not getting electrical signals through its nerves because its nerves have lost their identity. So what happens if we reset the clock in an old mouse, restore the ability to read the genes the way those nerves did when they were young? What happens? Well, we see that the electrical signals come back. The eye is still functional. And even though we only reverse the age in about half of the retina and the optic nerve, that's still sufficient for them to see again. A lot of the nerves in the eye are actually redundant. So here we've restored blindness during old age. I don't think anyone's done that before. Now, just finally, and I'll finish in a few more slides. This is the gene expression. So now we can look at all the genes in the back of the eye and ask what happens to those. And if we're right about this, we should see a restoration of youthful gene expression patterns. So here we've got what would happen if OSK treatment did nothing, right? Nothing happened here. If it made aging worse, it would look like this. This is where the genes, it would go up with OSK over here and up with aging. Now, if it reversed the aging process, it would go up with aging and it would reset. And those lines, those dots would fall along this diagonal. So that genes that went down a bit with aging would go up a bit with aging. And that's what we saw, that there was a nice reset of the gene expression pattern. And the type of genes are interesting too. You can see in red, there are many of these genes are involved in sensing chemicals in the environment. Some of them are even taste receptors that are in the eye or relatives of taste receptors. So that's super interesting that that's an aging effect that can be obviously reversed. All right, so what I've told you is that we think we've figured out one of the reasons, actually, if you include that cellular injury, two of the reasons that aging occurs. The main one that we've studied is a double-stranded DNA break in a chromosome, and that leads to a DNA damage signal. You repair the DNA. So remember, we do this in a way that doesn't cause mutations, but the act of repairing DNA leads to changes in developmental genes, among others, that leads to an old cell. Eventually, this cell could become senescent. We find that in our mice and particularly in cell culture, it accelerates the senescent process as well. We can also reverse aging for the first time. We can actually treat with these OSK reprogramming factors. We can contact the observer, reset the system, contact and find the backup copy of the information in the cell and reset it to where it can function again. And of course, we're currently testing whether a senescent cell can be reset as well. So what I've told you is that I believe that information loss is the main driver of aging, and that there's a backup copy of that information in every cell. Now, you might want to know, have we done anything beyond a nerve cell and a skin cell? The answer is yes. My colleagues around the world are now doing this in various tissues, in mice, of course, not humans yet, but spleen and liver and kidney can be reversed. We're doing muscle, it looks promising. And we're just ticking off the various tissues, but it doesn't look like that there's any reason why the entire body of a mouse and eventually a human could not be reset. And so that means one day we could go to the doctor, have a gene therapy or even a pill one day and take our age back a decade or two. And then the doctor will say, well, come back and see me in another decade or two, and we'll do it again. My lab is awesome. They fought bravely during COVID, came in, and I can't thank them enough for doing this research. This is Wancheng Lu, who is the first author of the December issue paper of his, which I'm very proud to say is the most downloaded paper at Nature for at least the last year. And I had a lot of help on the clock from, this is a bunch of people who are brilliant at understanding the clock. Patrick is a graduate student who's really taken up the clock into his PhD project, and he's just developed a method that we're trying to publish or going to publish soon that reduced the cost of doing one of those clock tests from a few $100s down to just a few dollars. And plenty of other people on the way. Alice has been major, Daniel and others. So often people ask me, what can you do? These are in the book. You can read it, page 304. This is what I try to do. I eat only one meal a day if I can help it. I try to exercise, do aerobics, try to keep my big muscles nice and fit. Hip hinges is, bending down and doing this with your body. I use biomarkers. I do blood tests a few times a year to make sure that what I'm doing is helpful. I wear bio trackers as well to see how I'm doing. Try to get a lot of sleep. Don't stress about life. It's not that bad. And we're all going to the same place eventually. And I try to eat plants as much as I can, even though I don't like them as much as meat. But I also try to eat plants that have been stressed out for reasons I told you. My father's doing well at 81. We're very proud of him, and he's a role model. And hopefully everybody in the world one day will have access to medicines that allow them to live and be productive and help with family and even start a new career in their 80s, which my father did. So that's the Nature paper from December and my book on the right, and all the collaborators that made this possible. So I've gone a little slower than usual, but I'd be happy to stop here. Thank you for having me, and I'll answer any questions you might have.
Well, thank you very much for Professor Sinclair to share the amazing view and perspectives on aging. And we believe we can finally conquer the aging one day. I think I'm not only one person very excited in the meeting, and there are many questions come from the chat box. There are really many of them. So I would like to deliver some of them for you. I will start with the most professional one and gradually go to more general ones. And more questions are welcome, and all audience can type the question into the chat box. So there's a question from the audience. Says, so the audience is a epi researcher working on longevity cohort study. He is asking, what is your perspective to apply these molecular markers like protein epigenomic or even transcriptomics as marker in population study?
Well, I think I understand the question. So there are various clocks. The DNA methylation one is the most popular, but you can measure immune age, you can measure glycation, which is various sugars being put on proteins. You can also measure frailty in animals and people and use machine learning to get numbers. You can do transcriptomics. You can do blood biomarkers, which is what I've been doing for over a decade. So those are very important. Part of the issue right now is that they're not done frequently in population studies. So I'm excited that it's becoming more and more routine. And that's one of the reasons that Patrick was excited about bringing the cost of that test down so that we could do millions of people's clock and then truly learn what's going on to drive aging and perhaps things that slow it down we could discover that way. But yeah, it's really important that we look at these other markers as well. Proteomics is a new one that was just published in the last year, that you can look at small proteins and large in the bloodstream and build a clock that way. Go back 10 years ago, 15 years ago, we had nothing. All we could do was just say, yeah, that animal or that person looks old and they don't walk very fast. But now we have these quantitative ways of measuring aging, which means that we can make much quicker progress in understanding how to delay it and slow it down and even reverse it.
Okay, thank you very much. And there's another question, I think slightly overlaid to my interest point, and I also would like to ask. As we have did a lot of research on OSK factor delivery, have we ever think about if we use adenovirus to deliver those OSK factors to, let's say, a larger tissue or like a larger mass organs, would that cause kind of unevenness, or would this kind of unevenness give potential dangers to the treatment?
Right. Well, that's what we're figuring out now. We did inject an AAV9, which is a type of AAV that infects many tissues, particularly the liver, of course. But we were thinking maybe we would get liver cancer in those mice, but we didn't. The mice were healthy. If anything, they had fewer tumors in their old age. So that's exciting, right? That it's not toxic if you deliver it intravenously. But there are some toxicities. If you turn on OSK in every cell in a mouse, which we have a mouse that can do that, it does lead to them having gut problems, and it will even kill them. So you don't want to turn it on at high levels in every cell. That part is for real. But if you do it in a targeted way that's low levels, and we also have an on-off switch so we can make sure that there is no toxicity, then I think it's going to be possible in people to treat. Now, whole body AAVs, it's still early days. We don't know how to evenly target tissues just yet, but there will come a time when we will be able to do that, and perhaps avoid the gut, which is where the issues seem to be in mice. So I think it will be possible, but it's early days. It's like asking someone who invented the first airplane, how soon will we have a Concorde jet? Well, obviously that's going to happen, but there's a lot more to do first.
Well, thank you very much. And yeah, I totally agree with you. And in my mind, probably even you just twist a certain amount of those cells to a younger stage, those cells probably will gradually replace those old cells in one day. Maybe it's a solution. Because I'm also doing some regeneration studies, and we use 3D printing to print living tissue and implant that into the mouse, see whether the repair functions. So maybe one of the direction in the future. So there's another question from the audience. I know Professor David Sinclair doesn't really research on telomerase, but our audience asking, can telomerase measurements be a milestone of aging?
Yes, they can. That part is not really debated. The question is, how accurate are they and how dynamic are they? And so I'm not the expert, but I do talk to a lot of experts. And it's somewhere in between, that you can get a representation of your age and your future health based on that. But it's not as accurate, is my understanding, as these other clocks. And we're learning that there are times when telomerase can actually be activated and rejuvenate some cells. Just while I think of it, there was a paper that, a couple of papers that have shown that it's possible to reverse the age of cells by lengthening telomeres. There was one that just showed hyperbaric oxygen, so high pressure oxygen treatment, apparently, according to that paper that was recent, does extend telomeres. So it is possible to regrow them. And the good news is at this point, it doesn't look like it's dangerous to do either. But it's always a worry because cancer cells love to have telomerase, and it might just be one step in the wrong direction.
Thank you. So another question. Audience wondering if all brain regions are equally affected by the aging. If not, what's the most affected region, and what tool we can use to promote understanding of how aging affects the brain?
Right. Well, it's not clear yet. People are doing now clocks on various parts of the brain and doing imaging. We can now do that even at the single cell level. As far as I know, the brain doesn't age differently according to the various clocks, but I could be wrong because we haven't finished the experiment. But we are looking in my lab, and others are doing this too, to look at various parts of the brain. I mean, clearly some parts of the brain break down earlier in people than others. The hippocampus is, a real problem for memory, and we know that breaks down. But I couldn't tell you if that was the clock of that part of the brain ages faster than another part. But what I can tell you that's interesting is that we are working on brain age resetting. We have the ability to infect the hippocampus in a mouse, and we've taken the ice mice that are aged rapidly, and we've restored their memory using our OSK system. And now we're doing just regular old mice, and that looks promising. Manuel Serrano has published a paper that he can do this too, and induced neurogenesis, so the birth of new neurons in the brain. So I think eventually we should be able to use this technology in the brain as well to restore old memories and the ability to remember.
Wow, that's really amazing, and we're looking forward to see that day. Another question for myself. So I did my bachelor and master's in pharmacology. So by reading your research on NAD and its precursors, NMN, well, it's really amazing, and the result is really amazing, and I think a lot of people benefit from it. I'm wondering, because just by calculation from the research, it's a pretty large amount of NMN needed for human. So, and this is the endogenous molecules. If we supplement it largely to body daily, would that cause kind of desensitization of body to NMN or its downstream factors, which leads to people need to have, all the mice need to have more and more over the years?
Yeah. Well, it doesn't seem so. We give the same dose to the mice throughout their old age, and they're less frail. And we've got early evidence, though we've got more mice on the way, that it extends lifespan. But that's just early days. I don't want you to tweet that one. But the point is that those mice are not losing their ability to turn NMN into NAD, as far as we can tell. In the clinical trials, we've done, well, at least a month of treatment. I think we've now got some 3-month data. Over that time frame in humans, NAD levels go up with NMN. If you take 1 gram or 2 grams a day, about 2-fold in the blood and in the blood cells, and they don't come back down. So I think, and we've done dogs as well, that's preliminary, but it looks like dogs maintain their NAD levels as well for a few months at least. So yeah, I don't think there's anything to worry about in that regard. What I think is more likely is that we need to time it right, because NAD levels are cycling during the day in the circadian rhythm. You don't want to mess up your sleep patterns. That can also apparently age you. But also we want to know how often do you need to take these NAD boosters. And as far as I know, nobody's tried that, even in a mouse, to try every other day or 2 times a week, just for lack of money and time. But I think those experiments would be great.
Thank you. I think it's very complex system in the body about of the desensitization, and I think a lot of the signaling are very robust and they won't be really affected by external agent. So there's audience asking, is there any clinical trial for MMN in the past few years? I think there are lots of them, but could you please briefly elaborate it?
Yeah, I mean, the ones I know the results of, there are lots of them that are being put on the clinicaltrials.org, but the trials that I've been affiliated with, and in full disclosure, it's a company I started called Metro Biotech. They have NMN being dosed to patients that have Friedreich's ataxia, a rare disease, neurological mitochondrial disorder. We also have trials in COVID, 30 hospitals, and then also for at the Brigham and Women's Hospital, they're testing muscle endurance and strength. And so those are the trials. We've been doing phase 1 safety studies for close to 3 years now in people with Brigham and Women's Hospital group, and have had no issues in those patients or those subjects. So that's all I have to report for now, but hopefully in a year from now, I'll have some good news or some bad news to report.
Well, we believe there are more and more research and clinical trial on it, and we're looking for good news, not bad news. There are a few very interesting questions, and I'm also confused about it, and I would like to ask. As you suggest, we shouldn't eat 3 meals a day. Does this mean we should fast for 18 hours every day and only eat during the 6 hours daily? So I guess he's more interesting, say, like, we don't eat 3 meals, but that's it. Like, you get my question.
Well, if you're an athlete or you're 20 years old, it doesn't matter. But if you're a sedentary 51-year-old, then it does matter. So I've, over the years, cut back on food. Initially, it was no breakfast. Then I skipped lunch. And now I skip both and have a small dinner, but not every day. I still like to eat out. I go out at least a couple of times a week for dinner, and I eat normally. I probably wouldn't order a giant dessert, but other than that, I eat normally. But I found that it's great for me, at least. It takes a few weeks to get used to not eating because it's a habit, for one, and your stomach and your brain go crazy at first. But if you want to try it, and I mean, please don't do malnutrition or starvation. That's not what we're talking about here. But it's restricting the time that you eat down to 6 hours, or in my case, half an hour. And so try it, do it for a few weeks to see how you do. Don't give up, because it takes a few weeks to get used to. Drink tea, drink coffee, that's fine. If you need to break the fast, eat an apple. It's not going to kill you. You don't have to be so draconian about it. Just try your best. And if you want, just skip 1 meal a day, either dinner or breakfast. And then you've got that period of sleeping without food and extending that. So why do I say that it's helpful? Well, it's pretty obvious. Calorie restriction and intermittent fasting is the best way to extend the health of an animal. And in places where people live longer, they also have periods of fasting as well. So it's not really up for debate that this is healthy, unless you overdo it. But make sure you're also getting enough vitamins and minerals. You don't want to be deficient in those.
Thank you. And well, I probably the one person who would like to reduce my calorie input simply because I would like to lose weight.
It's a very good way to lose weight. It really is.
Yeah.
I put on a bit of weight during COVID, and I've lost over 10 pounds just since February.
I'm very jealous. I would like to lose 10 pounds. I think I can like 20 pounds more over the COVID.
Yeah. Well, if you only eat one meal a day, I guarantee you're going to shed that weight.
Try it. So we are getting close to the time, but I think we just ask one more general questions. Well, that's a very interesting question. What are examples of stressed vegetables? So, because you use the word stressed vegetables, I think that's a very interesting description.
Yeah, it's good. This will be in my next book, one of the topics that I'm writing about, because it's so interesting and people want to hear about it. So red wine is the most obvious. The winemakers have realized over thousands of years that if you pick grapes that are on dehydrated vines or vines that are infected by a fungus or both, it produces very good wine, in part because they're filled with these molecules that I've talked about. Resveratrol is one, but all of these other colorful molecules, the intense reds, are part of the plant's defense. Other foods, well, I mean, if you can afford it, eat organic, particularly those plants that have been grown not in a greenhouse, if you can help it, or even if they've been in a local garden and there's been a caterpillar on it. Pests, too much sun, not enough water. These are things that make plants more nutritious, actually, and probably have more of these molecules. So I try to eat locally for that reason as well. And eat colored foods. Those colors are molecules that are usually associated with xenohermetins. So very dark green, the reds, the oranges, the purples, those are good. The opposite is take a lettuce that's been grown in California. It's almost white, it's barely green, it's all water, not very nutritious, and probably has very few of those xenohermetins.
Well, thank you very much for this. And well, I think somehow correlated with some Chinese medicine, the conventional one. Well, there are lots of dried and salted. So, well, and we will say usually the aged plant is better than the young one. Well, you know, the young one is full of water. They taste like fruit, not really a medicine. Well, there's really a lot of things we learned from this session, but we are running out of time. So it's very unfortunate we have to say goodbye at this point. I know most people like Professor Sinclair and would like to save him forever, but we really run out of time. So please allow me to express appreciation for Sinclair again to attend our summit, and looking forward to see the world that we can live to, say, 150 years old. Well, hope our summit is still there in the next 100 years. So very appreciate for that.
Oh, you're welcome. Thank you for having me today.